Welcome to this quick video on the nephron and ADH. But what is the nephron, you may ask? Well, the nephron is the functional unit of the kidney. It's the kidney's main, component of the kidney, and you have millions and millions of these times two, since we have two kidneys. And what the kidney does, as you guys might tell me, is that it filters the blood and helps us produce urine, in which we expel and excrete. But if you think about the word excretion, it's the removal of metabolic waste, and the most important word we're going to look at, and the most important wasteful metabolite, is urea. Okay, but first things first, let's draw our nephron. So represented in purple is our Bowman's capsule. Represented in red, because it's almost like blood vasculature, it's a vascular structure, it's our glomerulus. Okay. In purple is our what, guys? Represented by BC, Bowman's capsule. Indeed. So let's just dive into the Bowman's capsule and glomerulus, and remember, the blood has some wasteful structures, such as urea. So I'll do the mic drop moment in a minute. We need to filter these structures, and the way this is achieved is through high BP. So I'm using shorthand again. Patients check for the watt, where they put this particular medical instrument when you're in the GP, and it sort of squeezes and it gives you a reading in millimeters per mercury. Any ideas what I'm referring to? BP? Yes, exactly. High blood pressure. So by the way, if you're new to my videos, the upwards arrows obviously refer to high or increased. So you have high blood pressure. This high blood pressure achieved through the the renal arteries and the glomerular structure effectively will keyword small molecules through. Any ideas what those keywords, what keyword I'm looking for? And it shares the same, it's a unit. The newtons is the unit of what? Fantastic, force. So high blood pressure forces, and it's really important you say the word force. It forces small molecules. Now, what do I mean by small molecules? Well, small molecules such as, your micronutrients, your good old glucose, your water, because remember, trying to produce urine at the end of the day, urea, amino acids. Do not write amino acid because you're going to lose marks. But please do subscribe though. 1, 2, 3, 4, there's loads like ions, everything you guys have explored upon the IGCSE spec. So they're going to force small molecules from the glomerulus to the Bowman's capsule. So considering this is like an overview review video, I'm not diving into too much detail, but principally it's going from the glomerulus to the Bowman's capsule. Maybe after this video, once all my students have done their cycle test, I'll isolate ultrafiltration, selective reabsorption, and so on and so forth. So it forces the small molecules. What gets left behind? Yes, I'm asking you, one of my students, or one of my future students, or you guys, the YouTube community. You guys are my students. What gets left behind in the glomerulus, since it's too large? Exactly. So proteins are too large, so they get left behind. So it would imply, if proteins do end up in the urine, it would imply that something's gone wrong. There must be a disease. There must be kidney damage. There must be, kidney failure. A condition where this must have been damaged as such, or elevated levels of high blood pressure might have forced the proteins. But normally, in a healthy control / patient, this would not be the case, because the proteins are too large, so they get left behind. And essentially, this whole event of forcing the molecules through, this is known as the mic drop moment. Ultrafiltration is our answer. Okay, so we've done 1/2, well 1/3 actually. Now, Bowman's capsule is our endoglomerulus, are our first, substructures of the nephron. Let's talk about orange here, or purple, I don't know, I'm colourblind. Any ideas what this is called? It stands for the PCT, and the PCT stands for proximal convoluted tubule. Now remember it being proximal because it's next to the Bowman's capsule. The other one that's a bit further away is called distal, but we'll explore that later on. But anyways, the most important part really from the small molecules, list here is glucose. But my question to you, yes you, the viewer, why do we need to return glucose back into the blood? It's one of the good guys, but why do we need to bring it back? Yes, you're right. We're trying to maintain blood glucose level, but most importantly, rhymes with destination, it's used in respiration. We need to release ATP. We need to release energy. So we need to selectively reabsorb it. So glucose gets reabsorbed back into the blood. So that's what selective reabsorption is. So if I write in, let's pick a color. Yes, we'll click red again. Glucose is selectively reabsorbed back into the blood. It achieves this through a low to high cellular transport method. Any ideas what that might be? First word means, a synonym where you're online on a social media or, I don't know, application. Yep, active, and then the second word is a mode of what? Yeah, transport. So active transport is the way glucose returns back to the blood. So you can say selectively reabsorbed back into the blood via active transport. Fantastic. Quick fire question. I've got glucose over here. The arrows refer to what? Where am I? If they're going to be transported from the PCT, the third part of the nephron, and they're going to go where? Where they're going to get transported back into? Fantastic. The blood, via what transport? Low to high? Yeah, I'm talking to you? Yes, exactly. Via active transport. Now, there could be some follow-up questions on your cycle test or whatever test or mock exam or exam tomorrow where we could expand a bit more on active transport. The cells that line up these PCT structures could be, populated with tons and tons of mitochondria. So as you guys know, active transport, it says it on the tin, it's pretty active. It requires energy, ATP, to support that transfer window, really, from low to high. But it needs to be supported by tons and tons of energy, courtesy of the mitochondria. Also, there's a bunch of villi on these cells to increase surface area, so on and so forth. Brilliant. So this whole stage, if you could think of the PCT as the site of selective reabsorption. Okay, the next color and the next part, our next station on the nephron network is the loop of Henle. So I'm just going to draw an extended PCT, and then we've got the loop represented in blue here. So the loop of Henle. Now, luckily, guys, you don't need to know too much about this. It's where, osmosis happens here. We're turning the water back to the blood. But also there's a bit of salt maintenance, but that's very A-level. But for IGCSE, there's not much to it, to be honest. Just the fact that you've got a loop, loop of Henle, circle line. Next part is the DCT, so represented in another colour. Let's go for light blue. This is the DCT, and then we've got the collecting duct. Okay, these two respond to a hormone, which I'm going to talk about, but I'm only going to talk about it in the sense that, just like me right now, because I've had like one correct chai followed by a couple of coffees, I'm rather dehydrated. I'm only going to take it from that avenue. I'm not going to say when you're super hydrated. I'm going to leave it to you guys to think critically shortly after. So, DCT and collecting ducts are responsive to this 3-letter word. And I know you know it, but I'm now going to explain to someone who doesn't know it. Perhaps they've missed the lesson or their attendance is shocking. So, when you're really dehydrated, it's obvious that your water levels in the blood is reduced. What detects it? Well, our first key word is osmoreceptors. They detect, water concentration. So the square brackets refer to concentration. Where are they located? So there's a part of the brain which also controls body temperature. That's called the hypothalamus. That's the location. Underneath the hypothalamus, there's a structure, if I were to zoom in, called it stands for PG. Any ideas? It's referred to the master gland, a hormone responsible for secreting a plethora of hormones. Any ideas? Exactly, the pituitary gland. It's the pituitary gland that releases that 3-letter term, and that is ADH, antidiuretic hormone. And the letter H, hormone, means it travels through the what, guys? Exactly, travels via the bloodstream. So far, so good. So this part is just all talking about how do we get to ADH release. I'm dehydrated. Water levels in the blood are remarkably reduced. They detect this low concentration of water. These receptors called osmoreceptors, well done, they're located in the hypothalamus, and it contacts the pituitary gland to release ADH, which travels through the blood. Okay, but it's going to target two regent streets, and they are the collecting duct and the DCT. Now what does it do? Well, it makes the walls more permeable to water, so that more H2O is reabsorbed back into the blood. So if you're eating something salty, you need to, if you ever eat salty crisps or something that's rather salty, you're going to have low levels of, water in the blood. So as a consequence, ADH is released and your collecting duct wall is more permeable to water, so that water is more reabsorbed into the blood. By what process? High to lows, partially permeable membrane, begins with O, exactly, osmosis. But that makes sense, the DCT is more permeable, the walls are more permeable, collecting duct's more permeable. What does it mean in terms of urine? It's because urine's going this way, isn't it? Think about all the stuff that's toxic, like urea and those excess ions. My question to you is, what is the fate of urine? Put your hands up if you think urine is more dilute, since more of the water has returned back into the blood. Put your hands up if you think it's more dilute. Why are you putting your hands up? It's more concentrated. It's more concentrated because there's more excess ions, there's more urea, but less water. That's the principle here. And secondly, if more water is returned back into the blood, that means urine is lower in volume. If you're lowering the volume of water, if you're lowering the volume of urine, that means more water has been reabsorbed back into the blood. So if you think here, water's going back into the blood. Okay, so the fate of urine, put simply, if I can sort of squeeze it, where can I squeeze it here? Right here, makes urine more concentrated and it reduces the volume. Okay, Like I said, this is a quick overview. I'm going to make a couple isolating each part so that everyone has excellent fluency and understanding and comprehension of the material. But this is the Nephron and the ADH in a lesson format, almost, under 16 minutes. It's essentially like everything you need to know for the test, speed run, essentially, with a few more questions that I've asked you, the viewer, to get engaged. So yeah, please do like, comment and subscribe, and I'll see you in the next episode, and good luck in any future exam, past paper, topic test, and so on and so forth. Good luck, all the best, take care.